Hello, everyone. I thought it'd be good to do kind of a high-level overview of how our immune system deals with viruses like COVID. And I found this paper from 2016 in Seminars of Immunopathology that talks about that. So I thought that we would kind of peruse this paper together and go through some other things so that we have kind of a overview of how the immune system works in its various stages to fight off viruses. Okay. So before we get started, it's good to go over this tree of the types of the kind of the cast of players here. So on this side, we call the innate response cells. There's two kinds of two parts of the immune system. There is innate immunity and adaptive immunity, and this is a artificial divider that we've made as people that are studying it to help us understand it together, but there's a lot of crosstalk between the two systems, so they're not separated. The innate system is composed of these what are called granulocytes, eosinophils, basophils, and neutrophils, and then this thing called a monocyte that differentiates into a thing called a dendritic cell and a macrophage. So these things have things called granules, which are kind of like, you can think of them as like little weapons that they carry around, and they use these weapons to unload on pathogens to destroy them. And then this dendritic cell is kind of like a control center. It controls a lot of the immune system. And macrophages are, all these cells have many different functions, but this is kind of their basic function. Macrophages tend to eat things up and clean things up. And there's a thing called the natural killer cell, or often referred as an NK cell. This thing kills bacteria as well as your own cells that have been hijacked. And then these are lymphocytes, B cells and T cells. B cells make antibodies, and then T cells are effector cells that can kill cells and produce cytokines as well. Okay, so we'll come back to this, but it's kind of good to know the cast of characters. Before we get started on this paper, I found this website from Harvard Medical School, and they give a very good, quick overview of the immune system, so I thought we would go through that together. That way we can understand the paper a little bit better. So the first part is talking about this innate immunity, and this is our first line of defense. So when you first get exposed to a virus, the part of your immune system that's going to help you fight it off is immediately at the first exposure is the innate immunity system. And this system is always ready to react because it's always there. And then it's broadly specific. So these cells work on patterns that they see in viruses and bacteria that are not found in our own cells. So there's like these broad patterns that they have that are different than we have. And anytime they have molecules that respond to those patterns. And it's consistent, so it always reacts in the same way. So if you see the same thing over and over again, the innate immune system does not get better at fighting it. It fights it just the same whether it sees it for the first time or it's seen it 100 times. The innate immune system is made up of barrier tissues, cells, and molecules. Okay, so the first line of defense of your immune system is just a barrier. So dermatologists always like to say that the biggest organ is the skin. So your skin is a barrier. Your skin has a layer of this protein called keratin. That's the same kind of protein that's used in your fingernails and your hair is made out of. Your skin has a layer of keratin on it too, and that keratin is impenetrable. So that means if you get a virus on your skin here, it's not going to infect you. It's unless you have a cut or some kind of defect in your skin, the viruses cannot penetrate that. However, inside your nose and your mouth and in your GI tract and genital urinary tract, that's mucous membranes, and they don't have keratin, and viruses and bacteria can kind of go through those areas. The mucosa is protected by mucus. It has a thin layer of mucus on it, and in the mucus, there's a bunch of antimicrobial chemicals like defensins and things like that, and lysozyme and lactoferrin, all these little proteins that are in the mucus to try to kill the viruses and the bacteria that get in there. So that is a layer of defense. So the mucus protects you, and then there's these tight junctions that seal between the cells so that things can't get in between the cells. So basically, your first line of defense is just having a barrier. What happens is most of the viruses are stopped by this barrier, but if you have a ton of viruses, a few of them are going to make it across. So that's why viral load is important. If you're around someone really close and they just sneeze a bunch of virus into you, then it's more likely that a few of them are going to make it past this barrier and infect the tissues. Whereas if you have just a little bit, they're far away, you're in an outdoor environment, there's a lot of airflow, and you get a few viruses exposed to you, the chances that those few viruses make it through the barrier is much lower, just statistically speaking. All right. So the cells in the immune system include several types of circulating white blood cells. So let's go back to this. All these cells are called white blood cells. The lymphocytes, the granulocytes, the macrophages, the dendritic cells, the mast cells. These are all called white blood cells. So the major innate immunity cells are these cells. So you see the same things: neutrophils, monocytes, dendritic cells, macrophages, and mast cells. They use what's called, they use a lot of immune receptors that recognize common structures called pathogen-associated molecular patterns. So, I mean, that's just a fancy word that says that there's these certain patterns that are seen in viruses and in bacteria that we don't have in our own cells. And we have molecules that recognize these things. And these receptors are things like toll-like receptors. So you may hear, if you like to read papers, you'll see things called toll-like receptors, NOD receptors, RIG1 receptors. This RIG1 receptor basically identifies types of nucleotides nucleic acids that are not found in humans, like double-stranded RNA is not a thing that we have. And then AT-rich double-stranded DNA is not something we have generally. And this 5' PPP single-stranded RNA are things that we don't have but are found in viruses. They recognize those, and once they recognize those, they will send signals. So after recognizing a pathogen, the innate immune system does several things. First, they try to destroy it. So the neutrophils and the macrophages will try to eat it and destroy it. The neutrophils will degranulate, and then the dendritic cells will grab it, and the dendritic cell is like kind of the pathway to the adaptive immune system, which we'll talk about a little bit later. But basically, they'll grab it and process it and give it to the adaptive immune system. The mast cells and all these other cells, they release a bunch of cytokines. And you might have heard of cytokines like cytokine storm. They talked about that as one of the reasons that people die from COVID. But cytokines are this is a very simplified diagram, but they're basically small proteins that are used as communication molecules between all the cells of the immune system. They either activate or deactivate or tell them to do certain things. So yeah, then these natural killer cells, they get attracted to it and they will kill a virally infected cell. And then they release a bunch of chemokines and histamines, and they get released. And then they bring in a bunch of these things called complement as well as other cells. So here's the process. You get exposed to a virus. Most of them don't make it through the barrier. If a few do make it through the barrier, then these cells of the innate immune system will identify them as non-human. They'll be like, this is something that we're not supposed to have. They have certain molecular patterns, certain proteins and things that they recognize using these toll-like receptors, NOD receptors, RIG receptors, other kinds of receptors in the innate immune system that say this is not something that's supposed to be here. Then they start destroying it. And so if there's only a few, they probably will get destroyed before they even infect a cell. But then some of them will make it through and infect some cells. And when they're identified, they'll release a bunch of cytokines and chemokines. And these cytokines and chemokines activate the immune system and tell our immune system, hey, there's something going on in this area. You guys better come over here and check it out. So then all the white blood cells will kind of migrate into that area. And, like this says, there's these cytokines, they get into the endothelium of the blood cells and they make them kind of leaky so that cells can kind of get across from the blood. And that's why when you have an infection, things get swollen. It gets swollen because all your vessels are leaky and fluid is leaking out into the tissues, and it gets edematous and it gets swollen. So anyways, it'll bring in all the other cells, and then these things called complement, and they'll start trying to destroy these things. All right, so let's go to this part 2. All right, so these are the sentinel cells. These are the dendritic cells and the macrophages. These guys are going to try to find these viruses or bacteria and process them. And they recognize them using these toll-like receptors and things like that. And then they release these pro-inflammatory mediators. These are the cytokines. Then they get leaky, so like we talked about. Then the complement comes in. That's complement is a bunch of proteins. They're kind of like you can think of them as little machines that break apart bacteria and viruses. So they'll recognize the virus. They work in conjunction with antibodies, and they can also work without antibodies. And they will recognize viruses and bacteria and just basically tear them apart. Okay, so then this is where they make these cytokines make your blood vessel cells on the inside make these adhesion molecules so that white blood cells that are coming through will stick to them and say, hey, this is where we need you guys, and they'll stick here and they'll migrate across the blood vessel into the tissues. The neutrophils come in and they kill them. So they're eating them up and they're processing, and then they kind of commit suicide because they're like full of the bad stuff. Then when they come in, the macrophages will come in and clean up these cells. Yep, so here's the macrophages cleaning up those cells. And then if this, so sometimes people will get exposed to a virus, it'll make it past the barrier, and then the innate immune system will just take care of it, and that'll be the end of it. But sometimes it goes further, and the dendritic cells get involved, and they'll start presenting it to the adaptive immune system. Yeah, so what happens is, here's a dendritic cell. It got a peptide, so it has a little piece of the virus or the bacteria, and now it's going to go into the lymphatic system, and it goes in the lymphatic system and it goes to a lymph node. So lymph nodes are these things that are, when you get sick, you feel like these bumps in your neck or in your armpits or in your groin. These are lymph nodes, and in the lymph nodes, that's where the lymphocytes, they hang out in there. And then in the lymph nodes, they stimulate T cells of the adaptive immune system. So a T cell has this little receptor called a T cell receptor, and each T cell has a receptor that binds a specific thing. So in your body, there's all these T cells that can identify a bunch of things that you've never been exposed to. And so let's say you get exposed to a virus. It's a virus you've never seen. The dendritic cell will go into the lymph node and ask around and say, hey, do you guys recognize this thing? And a bunch of T cells will come up to it and be like, no, I don't recognize it. And then eventually it will run into a T cell that says, yup, my receptor fits that thing. And then that's when this dendritic cell will tell that T cell, hey, okay, so we need a lot more of you guys. So this naive T cell will start expanding. And that's when the adaptive immune system starts happening. So the adaptive immune system kind of kicks in around day 4 to 7. That's when it starts working. So up until about the first 4 days to 7 days, you're working on the innate immune system. Around day 4 to 7, the adaptive immune system starts kicking in. And the adaptive immune system is very specific. It will identify a very specific target, and it gets really good at destroying that. So it has specificity. It's very specific. It recognizes itself. So what you don't want is your immune system to attack you. So your body has ways of getting rid of the cells that attacks itself. So if a T cell has a T cell receptor that attacks your own proteins, your body will get rid of it, or it should get rid of it. When it doesn't get rid of it, that's when you run into what's called autoimmune diseases. So it recognizes itself, and then it has memory. So that means once you have seen this thing before, your adaptive immune system remembers it, and the next time it sees it, it will know exactly how to deal with it, and it'll deal with it much faster and much better. So the unique cells of the adaptive immune system are called lymphocytes. There are two types of lymphocytes. They're called T cells. They recognize protein fragments on surface proteins and kill the pathogen or help other immune cells. So see, this is the T cell receptor, and it recognizes this thing. So like, let's say that this is a virally infected cell. Viruses, what they want to do is so viruses are not alive. They're just little packets of nucleotides, either RNA or DNA, and they go into a cell and they hijack the factory of the cell and say, okay, you stop everything you're doing. Now all you're going to do is make copies of me. So it hijacks the cell and makes the cell just make copies of the virus and just make thousands of more viruses. Well, a T cell will come by, and this virally infected cell will on its surface show some of the proteins it's making. It'll be like, I'm making this thing. And if the thing that it's making is a normal thing that it's supposed to be making, the T cell will come by and be like, yep, that's fine, carry on. But if the virally infected cell is making viruses, it'll show a piece of the virus and say, I'm making this thing. And the T cell will come by, if that recognizes that, and say, yeah, that's a bad thing. You shouldn't be making that. And when it recognizes that, the T cell will tell that cell to die. It'll kill that cell. So basically it kills the factory, because that's what, then it shuts down production of that virus. Then B cells lead to production of antibodies, and then antibodies are these Y-shaped proteins that specifically recognize the protein or the peptide. So the antibody is a lot like this T cell receptor in that it binds the specific thing, except it's a free-floating thing. So it's not bound to the cell; it's a free-floating protein. And then when the antibody binds it, it can activate a bunch of the immune system. It can activate macrophages eating it, dendritic cells processing it, complement can destroy it. All kinds of things can happen with the antibody. Okay, so the main function of B cells is to produce antibodies, which it does, and then you can go back to one of our previous videos about monoclonal antibodies to show a little bit more about antibodies. And monoclonal antibodies were this amazing advancement in science. So yeah, we talked about this in that video. It's like a Y-shaped thing, and there's a variable domain right here that recognizes the thing, and then there's a constant domain. So they're talking about there's 5 major types of antibodies: IgA, IgE, IgD, IgG, and IgM. So IgM is usually the first type of antibody that's made, and then IgG is the most common. This is kind of the workhorse. This is found in mucosa. This is involved in allergies. Okay, so then antibodies just bind the virus, and basically when they bind it, when they talk about vaccines, they talked about neutralizing antibodies. A neutralizing antibody is an antibody that binds a virus in a place so that it no longer works. It binds it in the like the spike protein or something, so it doesn't work anymore. It can't infect anything, so it neutralizes the virus. There are non-neutralizing antibodies that bind the virus, but not in an important place, so the virus can still function even while the antibody is stuck to it. Those antibodies aren't worthless because the antibodies do activate other parts of the immune system to recognize that thing. Okay, so this is talking about complement. So once the antibody binds something, it activates complement, and the complement basically destroys it. Okay, this is a thing called opsonization. That basically means the antibody binding it makes it a target for macrophages and other phagocytes, cells that eat things, to eat it and destroy it. And there's another thing called antibody-dependent cellular cytotoxicity. That means that when an antibody binds a virally infected cell, a T cell or a natural killer cell can come and kill it. And they also mediate neonatal immunity. The IgGs of a mother can cross the placenta and be given to the child. So if the mother is immune to something, the child will also be immune to it for a while. The antibodies last like a few weeks. All right, let's go to the next one. Yeah, these I'll link this website. It's very good. The pictures are great. So here it is. This B cell. So B cells are just like T cells. They have a little B cell receptor, which is essentially a little antibody, and they have antibodies for almost every possible protein you could encounter in your life, things that you've never seen before. And when it recognizes it, it gets told that it needs to make more of it. So they develop in the bone marrow. So people think that it's called a B cell because it develops in the bone marrow, because that's where it develops in people. But it's actually called a B cell because it was originally found by these scientists who were working in chickens. And birds have this thing called the bursa fabricius, and that's where the B cell is in chickens. So it's actually called a B cell for the bursa fabricius, not for bone marrow. But in humans, it's developed in bone marrow. So they circulate. So after they get developed in the bone marrow, they end up in the lymph node, just like the T cells. And then the dendritic cell will bring it in here and say, "Hey, do any of you guys recognize this?" And then, if a B cell recognizes it, it says, "Okay, yeah, I recognize this." And then a T cell will signal the B cell to become activated and make more. And the T cell will tell them, "Hey, you need to make a bunch of antibody of this." And then when it does that, it becomes a plasma cell, and it goes to the germinal center, and it makes 1 billion copies of itself, a ton of copies. And so the B cell will become this thing called the plasma cell, and a plasma cell essentially is just a huge antibody factory, and it just starts pumping out a ton of antibodies and then just putting them all over your bloodstream. So now your body says, I've been infected by this virus. The dendritic cells have presented parts of the virus to these B cells, and several B cells will recognize different parts of this virus and pump out a ton of antibodies about it that recognize this virus. So then these viruses go through everywhere, and they start attacking things. Okay, yeah. Oh, and so this is another cool thing that we talked about in the monoclonal antibody video. But basically, after the B cell recognizes a thing, it undergoes something called somatic hypermutation. What happens is it slightly changes the binding site, this little tip here, and it tries out all kinds of little minor changes and sees if any of them make it bind better. And if it does, that one becomes selected to become the dominant plasma cell. And also the important thing is some of them become these things called memory B cells. And memory B cells, they don't actually make antibody, but they just hang around, and they're ready for the next time that you get exposed to the same thing. So this is what happens with a vaccine and with natural immunity. If you've been exposed to a virus before, or you've been immunized against the virus, you have these memory B cells. And when you get to see the same thing again, it's ready. It says, I recognize this one. I know exactly how to deal with it. And it very quickly ramps up the immune response and makes a ton of antibody for that. Okay, so this is the long-lived, and then these are the memory. These will become reactivated. They'll expand themselves and make more plasma cells in the future. Yep. So now you have these antibodies floating around. So like after you've been infected or after you've been vaccinated, you have a lot of long-lived plasma cells and memory B cells, and these will circulate some antibodies in your blood all the time at a kind of a lower level so that you have it. And that's why, after you have it, people can measure antibodies against the virus for months afterward. Okay, so then we're talking about a little bit about T cells. So same thing. They talk about all these things. So T cells, instead of being in the bone, they go to the thymus. So they start off in the bone. They go to the thymus to get matured, and then after they get matured, they end up in the lymph node. Same thing, and that's when a dendritic cell or an antigen-presenting cell will show a peptide and ask them if anyone recognizes it. Then they become either CD4 or CD8. So CD4 are often called helper T cells. These are the ones that are infected by the HIV virus. So helper T cells are infected by the HIV virus, and that's how HIV kind of hurts your immune system, by knocking off the helper T cell. And then the CD8 are called killer T cells, basically. But they have lots of different functions. And this is a very simplified explanation of how the immune system works. And also, we honestly don't know exactly how the immune system works. This is how we think the immune system works, and there's a lot of experimental data that suggests that this is how it works. And we have a pretty good understanding of most of this, but there's a lot of complexity, and this is a very simplified explanation of the immune system. And I think that's one of the things that kind of a pet peeve of mine, because people think that science gives you facts. Science doesn't give you facts. Science is a process, and science is a way of understanding the world better. But it doesn't give you a set of facts. These are not facts. These are just what we think, how it works, and it's a kind of a probability statement based on experiments. Okay, so starting with the dendritic cell again, it goes to the lymph node, activates these T cells. And they're just saying that the T cell has to be given antigen through this thing called the MHC, which is called major histocompatibility complex. That just tells you that this is a friend that's giving it to you and saying, hey, check this out. If it doesn't have an MHC, then it won't respond to it. So once activated, the naive T cell, so this naive T cell has never seen this antigen before, but now it's seen it. It's like, hey, it's my time to shine, and I'm going to make a ton of soldiers. Then after it's made a million copies of itself, it starts leaving. And that's why you notice that the lymph nodes get bigger when you're fighting an infection. That's because all these T cells and B cells are in the lymph nodes multiplying like crazy, getting ready to go to war, basically. So that you can think of like the lymph nodes as the barracks of your army, and they are making more soldiers to go fight. They're called cytotoxic T cells, or sometimes they call them killer T cells. They travel throughout the body, recognize cells that are presenting the peptide. So if there's a virally infected cell, they're presenting this viral piece saying, hey, I'm making this stuff. The T cell says, nope, you shouldn't be making that, so you better shut down. You better die. So then they'll kill it, and then macrophages will come by and clean up the dead cells. These are called helper T cells. They just produce a bunch of cytokines. And the Th1 cells activate macrophages, and THC cells activate eosinophils to kill parasites. So if you get a bunch of Th2 helper T cells, you get allergies. So and there's a whole nother thing that maybe I'll make a video on about the hygiene theory for allergies. Okay, so yeah, there's a whole bunch of different T cells. We probably don't have time to get into all of these. There's the follicular helper T cells. They make the B cells work better. This Th17 produces inflammatory responses, produces a lot of cytokines to bring in neutrophils and things. And then there's this thing called regulatory T cells, or Tregs, and these guys basically suppress the immune system a little bit. And you're like, why would you want to suppress the immune system? Because if you have a runaway immune system, then you get too much inflammation, and that can be bad for you. And also you can get autoimmunity, and that's bad for you too. So there's a lot of cells that suppress the immune system, like regulatory T cells, and there's this thing called myeloid-derived suppressor cells. They're kind of like macrophages that suppress the immune system. So the immune system is you need a balance. You can't have like a super activated immune system, because that would be bad. Although initially that sounds good to have a super activated immune system, but that's not good either. You need to have a perfectly regulated immune system. And so after a T cell response, there's a memory T cell. So this is what we talk about too. After you've been infected with a virus or with a vaccine, you will develop memory T cells that respond much more quickly to future pathogens. Okay, so now we kind of have a basic idea of how the immune system works. So let's quickly peruse this paper. So they're talking about, in this part, they talk about direct viral injury. So they're saying in this paragraph that there's direct viral injury because the viruses take over cells and they make them produce a bunch of virus. And a lot of times, the viruses make the cells pop, and those are called cytolytic viruses, or otherwise it your body stops functioning normally because those cells are just making virus and not doing their normal job. However, this is the important thing, much of the clinical sequelae and damage to respiratory cells is the result of the host response. So in most of these viruses, it's not the virus itself that causes all the problems, like the lung damage and being on a ventilator and having GI problems and all kinds of stuff like that. That's not what's causing the damage to you. It is the response. It's your immune response. So virus sensing, now this is talking about those PAMPs again, those pathogen-associated molecular patterns. There's these pattern recognition receptors we talked about, like the toll light receptors, RIG1 receptors, nod receptors. We talked about them. So this is part of the innate immune system. Then what the cells do once they have these pattern recognition receptors activated, they'll make interferon. Interferon is like the first cytokine that your body will make in response to a virus, and the interferon activates the immune system to bring in cells, so it'll bring in a bunch of cells. And also interferons will start having your body make a bunch of antiviral proteins within them. There's these proteins that cut up RNA inside the cell, which you don't normally want a lot of because you make RNA as well. But in the setting of viral infection, it'll make this protein that chops it up. It's called RNase L. And then there's all these proteins that they make that bind up the viral capsid, the coating of the virus. It'll lock it into the cells. So anyway, your body will start producing a bunch of stuff that knocks down viral production within the cell in response to interferons. So yeah, they're talking about how the epithelial cells, these are the cells lining the nose and the lungs and things like that, they'll start producing a bunch of cytokines too. And chemokines are like cytokines, except they just attract white blood cells to come there, the neutrophils and things. So this is talking about things like lysozyme, lactoferrin, and secretory leukocyte proteinase inhibitor. These are those things that are in the barrier, in the mucosa, and these things will kind of neutralize a lot of the viruses. And there's other things called like defensins. Oh yeah, here they are. Beta defensins that neutralize antibodies. And these are the things that like one of the things they talked about in COVID is how vitamin D is important. Vitamin D is known to help bind receptors in the nuclei of cells that coat and help them produce these beta defensins and lecithins. Wow, say that 9 times fast. And it makes these things. So if you have low levels of vitamin D, then you have a hard time making these, and it'll just you won't be doing as well. And it's involved in a lot of other receptors within the immune system. So they talk about endothelial cells. We saw them. They get kind of leaky, and they let complement and macrophages and things come in. Now they're talking about the neutrophils. So once they bind them, they release their granules, and also they make these things called ROS, which is called reactive oxygen species. These are basically oxygen is a pretty reactive atom, and they'll make these molecules with different kinds of oxygen in them that are very reactive, and they basically, you can think of it as they corrode the proteins of the viruses and the bacteria. And then they're talking about the NK cells, just like we saw in that slide deck. They're talking about the ADCC, again, the antibody-dependent cytotoxicity. Basically, they will identify the virally infected cells, and the natural killer cells will kill them. Okay, so I think this is basically a lot of repeat of what we went through in these slide decks. But I think if you want to read this paper, you'll have enough information to kind of get a lot of some stuff out of it. But basically, initially you have, to sum it up, when you get exposed to a virus, most of them won't make it past the barrier. There's a lot of proteins that stop them in the mucous membranes and in the mucus that's lining the mucous membranes. And if a few make it through, you have these pattern recognizing receptors that will recognize them, bring in macrophages and neutrophils to try to kill them, and that's the innate immune system. A lot of the viruses won't make it past the innate immune system. If they do make it past them, then the dendritic cells will go to the lymph nodes and shop around the antigens that it finds and find the B cells and T cells that recognize it. They will then produce a lot of soldiers that make antibodies and cytotoxic T cells, and then they will spread throughout the body and eliminate the virus. Now, the problem is that sometimes this gets too robust, and there's way too much cytokines being released, and there's so much inflammation that it causes tissue damage in the lungs, and then the lungs stop functioning. Then they don't process oxygen well, and then you have problems where you end up on a vent and have problems. And that's why a lot of the treatments are basically aimed at decreasing your immune response from being overly aggressive, like they're giving dexamethasone. And this Anakira was one of the other medications, and fluvoxamine is another drug that they're trying that reduces kind of the hyperimmune response to it. And then the other things are these monoclonal antibodies, which basically gives you an adaptive immune system response way earlier. So instead of waiting 4 to 7 days before yours kicks in, if they try to give it to you earlier, then it can help clear the virus a little bit faster. Okay, so I hope I will link this paper and this Harvard Medical School site in the description below. I hope this was a little helpful to everyone in understanding how the immune system deals with viruses, and maybe some of the things that you read will make a little more sense with this background. So thanks for watching. If you have any questions or comments, please put them below.